for adhering to the guidelines. Good. So we are talking about the LIDISKI project. It's EU support, European Union support to livestock disease surveillance and knowledge integration. So we are looking at two major diseases. That is participatory PPR in sheep and goats, and then Newcastle disease in poultry. And we have four major partners. We have CIRAD, we have IZSVE in Italy, CIRAD is in France, and we have NVRI in Nigeria, the National Institute of, the NVRI, National Veterinary Research Institute, and then we have ICORE International Development Limited. Now the program is for 5 years and we have 4 major actions. The objective of the project is to integrate knowledge collected from actors in the animal health sector to provide tools and know-how to implement sustainable surveillance and control strategies against 2 livestock diseases, participatory PPR in sheep and goat, and Newcastle disease in poultry. Now, the context: smallholder food security heavily relies on their livestock as critical assets, coping with shocks, and exchange for income, amongst other things. Now, Nigeria's agricultural sector is highly vulnerable to environmental changes. There are due to large parts in semi-arid northern parts of Nigeria. So many of these smallholder farmers are largely in the northern part of Nigeria, and there is more mobility of animal population. Now, livestock diseases and cost of management is the biggest economic threat to livelihoods of poor livestock owners. Now, the central principle for sustainable surveillance and control of diseases to improve commitment of local actors through capacity building and early engagement in the development of the strategies, and then to develop strategies that are tailored to the context and to actors' expectations. The use of participatory approaches, integrating local culture and knowledge and socioeconomic context. Now, the LIDISKI project is centered on 4 major actions. That's capacity building of animal health actors, then engaging communities in surveillance and control, understanding the epidemiological and socioeconomic context. That's the 3rd action. And the 4th action is developing tools and guidelines from all the activities on the field and from every finding and discovery. So we develop tools and guidelines and scale up strategies. Now, target group is animal health actors. This includes public and private vets, community animal health workers, vaccine producers, and suppliers. Now, the final beneficiaries are the livestock smallholders. So we also have associates, they are listed there. Now, quickly, without taking much of our time, we are in the last phase of the LIDISKI project, and this is where we share findings from researches conducted during the project. And this brings us to the finding on the new protocol for diagnosing Newcastle disease in Nigeria and other West African countries where the strain is being discovered. So I'll hand over now quickly to Isabella to take it up from there. Isabella, the floor is yours.
Thank you very much, Tommy, for the kind introduction, and thank you very much for organizing this meeting, which I think is a perfect opportunity to discuss about diagnosis of Newcastle disease, which is a very important disease for animal health and also food security. So I start sharing my screen. Please let me know if you can see my monitor. Is it fine?
Yes, we can see your screen.
Okay, perfect. So My presentation today will be divided in two main parts. So I will briefly describe the disease itself, because I know that there are people attending that are students new to the disease. And then I will focus on the diagnosis, and in particular on the description of this new molecular tool that we developed in the context of the European project named LIDISKI, that Gesù Tomi has kindly introduced and presented. So let's start from the importance of the disease. Why is so important, Newcastle disease? First of all, because it is clinically relevant. This virus can cause a disease that is very similar to the one caused by highly pathogenic avian influenza viruses, viruses that are in a certain meaning overshadowing Newcastle disease because it's widespread at global level now. Newcastle disease, as well as influenza, they both have a very wide host range, and these properties is also a cause of concern because it can cause, let's say, challenges in the control of the disease because you have too many hosts to control. In addition, it can spread quickly, and this can result in enormous economic impact. In addition, we all need to remember that this is also a virus that can cause zoonotic infection, generally just mild infection in the eyes, so conjunctivitis. But remember that recently, through metagenomic approaches, there have been the identification of severe infection in immunocompromised patients with neurological clinical signs. So for all these reasons, this virus is included in the WOA listed disease, and it needs to be promptly reported. However, reporting is not that easy because this virus is impressively difficult to be diagnosed. We do not have pathognomonic clinical signs. We do have a lot of diversity in terms of virulence. We do have a lot of genetic diversity that render challenging even molecular development of new tests, as you are going to understand through my presentation today. So what caused the disease? The disease is caused by an RNA virus, so it's highly variable, as expected by an RNA virus itself. And in particular, behind the disease, there's a virus that is called avian paramyxovirus type 1. Recently, the International Committee of Taxonomy for Viruses has changed the name of this virus. So now we call it avian ortholavulavirus type 1 or orthoavulavirus javanis. Across my presentation, I will keep the former name avian paramyxovirus type 1 because this is what the literature is still doing, maintaining the original name. There's a lot of diversity among the avian paramyxoviruses type 1. First of all, in terms of virulence, there's this test which is called intracerebral pathogenicity index, and you can see here the cheek that is highlighting which is the target of this test, an injection that is followed by the observation of the animal health status. According to the results of this investigation, you can assign the different viruses to different categories: lentogenic, when the viruses are not aggressive; mesogenic, when they demonstrate a mild infection; and velogenic, which are the one we need to report to WOA, which are the one really aggressive with a high intracerebral pathogenicity index, as they cause a serious disease and they cause death of most of the chicks infected. In addition, we can distinguish the different viruses according to their tropism. So it means, sorry, I can see that there's people asking to enter in the meeting. So I asked Gesù Tomi, please be sure that all the people asking to enter are properly accepted. So sorry for the interruption, just a logistical information. By the way, we can describe this virus also in terms of different tropism. What does it mean? It means that you can infect experimentally specific pathogen-free chickens and see that the viruses can have a very distinct tropism in terms of organs. So we do recognize velogenic strain, which are viscerotropic, so they have a tropism for the gastrointestinal tract. Neurotropic, it means that they go and infect the brain mainly, and the nervous side are the more evident clinical signs. Then we do have mesogenic and lentogenic and asymptomatic enteric disease, infection. These kind of strains, the ones which are not causing serious diseases, are the ones that generally are used for vaccination. There's a lot of diversity also in terms of genetic, and this renders very difficult the diagnosis. We do have recognized two classes of avian paramyxoviruses. Class 1, which is composed by just one genotype, and class 2, which is highly variable and contains more than 21 genotypes now. This is the very recent, let's say, nomenclature that has been generated in a work that describe the genetic diversity for the gene F, so the fusion protein, one of the major glycoproteins which are present on the surface of the virus. So this system was used to describe the diversity, and as you can see, you have here the phylogeny for class 1 viruses, and here you do have class 2 viruses. And as from the color, you can see there's a lot of distinct genotype inside. What does it mean? It means that we do have a single serotype, serotype 1, but there is a great number of genetic variants of this virus. It's like thinking about SARS-CoV-2, the COVID-19, we had many variants. And this is exactly what happened with this virus, and even worse because the diversity is much more evident. How this virus can be transmitted? Well, the virus is shed during the incubation period, during clinical stages, and for a limited time, even during convalescence. So we need to be very careful when we introduce new animals inside a farm or inside our house and our backyard poultry. We need to remember that the virus can be transmitted by direct contact, as expected, but even indirect contact. And for this reason, I paste here the picture prepared by, in the framework of the LIDISKI project, that is highlighting how also human beings can be responsible for the spread of the virus, and not just the animal directly. It's a disease that we know since early 19th century, but this disease is very difficult to eradicate. And reporting is, let's say, a global issue. And there's actually underreporting of this disease, also for the diagnostic challenges I was referring to earlier. So what we need to look for to identify this virus? First of all, we need to look for nervous signs. This is the most frequent evidence of infection of Newcastle disease, despite this is not pathognomonic. Generally, the twisting of the head, as you can see in this picture, is a very common sign with Newcastle disease, the neurotropic viruses clearly. But sometimes the signs are respiratory, and you can see animal gasping for air. The other signs you can observe in your flock is diarrhea, greenish, watery diarrhea. And there's also the possibility that you can find all your animal dead without any kind of clinical disease appearing earlier, like a poisoning. And the other option is changing the color of the eggs. The gross lesions are quite interesting because they are very similar to the one caused by avian influenza, except for this proventricular glandular region that is generally very much affected by viscerotropic Newcastle disease. There's then peritonitis you can observe, and even necrotic hemorrhagic lesions. So all these manifestations, as I was saying, really resemble highly pathogenic avian influenza viruses. So what does it mean? It means that if you are suspecting the presence of Newcastle disease, you need the laboratory in order to understand what is in front of you. And there's no diagnosis possible based on clinical signs. You need to collect the samples, and which kind of sample are recommended? Well, it depends. If you have live birds, you can conduct swabs. Tracheal and cloacal swabs are the one I mostly recommend. From that birds, there's a long list of organs that can be infected by the virus. But in addition, if you are in difficulties during necropsy, also considering the differential diagnosis with influenza, which is also a zoonotic disease, you should consider the possibility to collect cloacal swabs and tracheal swabs. So how we do the diagnosis of this disease? Well, from the laboratory perspective, I know that most of the laboratory now are equipped with real-time PCR machine, which are really useful in order to implement the diagnostic process. There's a screening method that you need to apply, first of all, and I've included in these slides that I'm going to share with you the link to the protocols that we use here as a WOA-FAO reference laboratory. We have been developing a very detailed description of the procedure available. So we do have to apply a screening method, first of all, and we here use this protocol from 2019. It is very sensitive. It is targeting the L gene, which is a conserved region of the Newcastle disease virus. It doesn't give you information about the pathogenicity of the virus. So it's just a screening method, a starting point. And if you have positive results, you can move on with characterization, which is what you need in order to understand if you are in front of a notifiable Newcastle disease or avian paramyxovirus type 1 virus. It's very important to not stop with the screening method. So how to type these viruses? Traditionally, you can use an in vivo test, the one I was describing earlier, the intracerebral pathogenicity index. The virus is considered highly virulent and notifiable to WOA when it has a value higher than 0.7. The problem is that this test is difficult to be implemented. You need to have the facility to do it with biosecurity measure applied. It's time consuming because you have the observation time, which lasts for long, and you cannot wait for long with a virus that is spreading so fast as it is Newcastle disease virus. In addition, you have the ethical issues that you need to consider. I mean, these animals can be food, can be a lot of things for us, and it's a pity to use them for a test. The other option is to use molecular pathotyping. Molecular pathotyping here is implemented using the test you can see here, and you can find the link once again with the description of the protocols, and I will give you the presentation as a PDF so you can absolutely download everything. And in case this molecular pathotyping method is positive, you can also use the amplicon in order to be sequenced. When you do have the sequence, you know that a virulent strain are recognized because they have in a specific position of the GNF protein, a leucine, together with few amino acid, basic amino acid. So it's 3 generally or lower for a virulent strain, sorry. For virulent strains, they are more than 3. And in addition, you find a phenylalanine in position 117 in the alignment. I know that many of you are not using sequencing, so I know that this can be not easy to be followed. And for this reason, we decided also to, let's say, develop a new tool for doing the diagnosis. So the drawback of these methods is that there's no sequencing technologies in many countries. It's still a major issue. And in addition, the maintenance of the sequencing technology is not that easy. In addition, this test may lack sensitivity and is not able to detect the co-infection, which is something in particular where vaccination is used, very important, because if you find a positive test using the screening method, you do not know if it is a vaccine strain or if it is a virulent strain. On this background, we decided to develop this test, this new molecular pathotyping test, which has been recently published, and I will provide you with the information in order to retrieve the procedure in the public database. So what's new? What is characterizing this new protocol that we developed in the context of the LIDISKI project? First of all, it is a real-time PCR, so not a conventional PCR, and then it's fast. In the second, you can do a recognition of virulent and avirulent viruses in dual mode. What does it mean? It means that you have a real-time that is providing probes that recognize virulent and avirulent strain according to the diverse dyes that are marking our probes. In addition, the product of this real-time PCR can be also sequenced. So still you have the possibility to use the conventional approach, which is the one recommended by WOR. So do sequencing. You can take your amplicon and send it outside, but you can also do a real-time to recognize virulent and avirulent strain. Let me better describe how we got to this result. So to set up this real-time PCR, the first thing to do was to align a very, very long number of sequences in order to recognize primers and probes to recognize the diversity. So we decided to keep separate sets because we wanted to have sensitive tests and able to recognize the diversity. We start with set A, that is composed by 2 common primers, and inside you have a probe for a virulent strain and a red dye for virulent strain. So this group of viruses, which are listed in this phylogeny, are the one recognized by set A. We move on with set B, which is much more inclusive because you can see that it includes all the viruses that are clustering in this very big pink colored part of the phylogeny. And even in this case, we do have common primers and then 2 distinct probes because we want to recognize the avirulent and virulent strains. The last set we designed was the one for the class 1 viruses, which are the one on the right side of the screen, the class 1. There's less diversity and they are generally avirulent. So you can keep these 3 sets together or decide to privilege one of them according to the epidemiological scenario you are facing in your country. So let me give you an example. If you know that in your country you are facing, because you characterized it earlier, the introduction of a genotype 7, you can maybe work mainly with set B. Despite my suggestion is to include both the set, because in this way you can also recognize eventually an introduction of new strains. How does it work? Well, here with the help of my colleague Andrea Fortin, who is here with me and who is the one who has been developing and validating and do a lot of work in order to go to these results, we have here 3 distinct masks that are describing how we need to prepare the mixer for the distinct set of primers and probes. So as you can see, we have here the set A with the distinct probes and the distinct class A, class 2A set primers. Here you have the set B and the probes that are used, and you can see here the 2 distinct probes for the virulent strains, and then the last one, which is the class 1 C set. So just a couple of things to highlight about this distinct mix that you can prepare. They do have different concentration, different final concentration. So be sure to follow the instruction that you can see here in my talk that I'm going to share with all the group. There's another important aspect, which thermal profile to be used for this real time. Well, we do have different annealing temperature corresponding to 55 degrees for set B and C, and we do have 60 degrees for set A. It's important to highlight that the possibility to merge all the sets together depends from the instrument that you do have in your facilities. If you have a real-time PCR that is able to have different temperature in the same run, you can clearly run all the sets together. If not, as it is the case of the BioRad instrument that I know is pretty common in many countries as mine, you can run set B and set C together, and in a separate plate, in case of negative results for this test, you can run the set A. My suggestion is to prioritize set B and set C because these are the sets including the virulent strain. So that's then the aspect of the ramp rate. This is very important. We demonstrated that the change in the temperature for the annealing can have an impact on the sensitivity of the machine, of the protocol. And I'm going to show you it later. So my suggestion is to use an approach decreasing the speed of temperature to 1.6 degrees per sec. This allows the annealing to occur much better and increase the sensitivity of the test. This nice slide has been also prepared by Andrea, who is here with me, and you can see that this is the interpretation you may need when you read the results of this real time. So you will have the green fluorescence, which is characterized the FAM probe, which is the virulent one, and you have the red light for CY5, so for the dye for the virulent strain in all the sets. So if you find a situation when upon your threshold you find the green, it means that your strain is a virulent, not aggressive, mainly a vaccine strain. If you find the red light, you need to be, like say, red light. So be worried because there could be a virulent strain inside your sample. And so the red color is the dangerous one. And remember that this protocol has the advantage to recognize also co-infection. And in a country that vaccinates, and this is very common, my country is vaccinating, and I'm sure that many or most of you are also using vaccine, you can recognize in case there's co-circulation of the vaccine with a virulent strain. And this is really useful. But there's another aspect positive about this protocol. You can also do sequencing of what you obtain applying the distinct set. So if you have a positive results, you can keep the amplicon and use it to run it on a gel to see that the dimension of the distinct set is enough big to be sequenced by a Sanger approach. So you prepare a dilution 1.1 to 5, and then you can run on a Sanger sequencing machine your results and confirm even the results of the real time. We have been doing an extensive validation of this protocol, and all the sensitivity tests have been run in parallel with the screening method. We know very well that screening method is more sensitive. This is expected because this protocol is a typing method that you need to apply downstream to the screening method, which is in this case the Sutton protocol I was mentioning at the beginning of my talk. We also test the analytical specificity of this test, and we verify, first of all, the inclusivity, so the test can recognize properly the different genotypes. And in addition, there was no cross-reaction with matrix components or non-target viruses, such as infectious bronchitis virus or avian influenza. Then the most important part, and for this part it was essential, the contribution of many countries, and I'm really grateful to all the colleagues that participated in this project. We have been testing samples which were negative for avian paramyxoviruses tested through other procedure, and 59 positive sample of different genotype coming from different countries. And with them, pathotype, the pathotyping approach was compared to the Sutton one. Let me briefly interpret this graph that I can understand is quite difficult to be interpreted. Here you have the CT value we found for the Sutton protocol, so the screening method. Here you have the CT value we identified for the set A and B, which are the most important one because we recognize the virulent strains. Well, you can see that generally there's agreement between Sutton and the set A and B, but there were a few situations where the set A and B were less sensitive than Sutton. What does it mean? It means that even with high CT value, it could be that you do have a real positive. And so be careful when you set the threshold, or better, not the threshold, the cutoff value for your procedure. Clearly, all the amplicon can be sequenced. So if you have access to a sequencing machine, consider that in our case, in our test, most of the amplicons we obtained from the real time were successfully sequenced and proper to pathotype, so long enough to recognize which kind of virus was in front of us. So before closing, remember that also we have been inviting distinct laboratories, FLI and National Veterinary Institute in Sweden, to try running the test in their own facilities. This was very important also to see how different machine, different extraction protocol can influence the sensitivity of the test. And actually we found diversity. So even if the diversity was not significant, it was evident that there was a CT value that was different across the distinct laboratory. And in one lab in particular, they did a comparison using a different ramp rate in the instrument. So changing how fast the temperature for annealing was changing. And they demonstrated that if you use as lower rates, the one I was mentioning earlier, 1.6 degrees per second, you obtain a better sensitivity, as you can see here. So before closing, just few recommendations. You can use this test, you can order primers and probe, or you can ask to these probes and primers to be made to make available them in some way. But remember that before using it, you need to optimize it, because in case you are changing reagents and equipment, you need to be sure that the test work as expected. So be ready to do comparison. In addition, you need to remember that even high CT value can be indicative of infection. So my suggestion, our suggestion, is to find a way to sequence samples which present CT value higher than 35 CT. This is because you can have in front of you a new virus that has different reactivity. So remember that sequencing still is important. You can use this test as a DIVA strategy. So if you have vaccinated animal to be recognized, you can implement the different probes to recognize virulent and avirulent strain. And remember that we were not able to, let's say, test the analysis on a large number of American strains. So it could be that this genotype may be not visible. I mean, it may be the ability for the test to recognize them is not the best one. We have the in silico evaluation clearly, but it's another story when you test them indirectly. And remember that you constantly need to check the oligonucleotides, because it's important that you monitor the situation in the databases in terms of new sequences published. I close the presentation just showing you the tool that is available online. You can download it anytime the complete description of the protocol. And I really thank the LIDISKI project for giving us the chance to work on Newcastle disease, and in particular on the development of this specific protocol. And I want to thank colleagues who participated providing samples. This was instrumental to get to the final results, to have a new test for all of us. And I close my presentation here, and I thank you, and I invite you to contact us in case you may need any additional information. Over. So I leave the stage to Jesus Tommy, maybe.
Okay, thank you, Isabella. That was really expository. Thank you very much. I don't know if any of the participants have a question for Isabella before we leave.
Yes, if anyone has question, we would be happy to assist you. Even you can use my email address in case you are running out late and you cannot stay here for long for the discussion, or if you are not, in the condition now to think about questions you may have on the protocol. And but really, my suggestion is to try finding a way to test this option, because I know sequencing is expensive, and also buying probes, which are needed for this real time, is not coming for cheap. But clearly, having a tool like this allow you to test many samples in one run, and so it could be useful to try finding the way to control a bit better this disease, which is really a plug for all of us. Over.
Thank you very much, Isabella. And thank you for everyone who has dropped a comment. You can always send your questions, like Isabella said. We'll put our email address in the chat box, and my own as well. So if you want to send your questions, you can always reach us. Also, this video will be available on YouTube. So you can always revisit this each time you want to check the protocol again. You can always refer to it, you can always share to a friend, and you can very well subscribe, okay? Subscribe to our YouTube channel. Thank you very much for your participation. Any questions before we leave? Any hands up? No hands, I guess. And no question in the chat box. Okay. Thank you very much, everyone, for your participation. Feel free to reach out to us. Thank you very much, Isabella, for being here. And thank you to your partner there. Thank you very much for being here. All right.
Thank you so much. It was a pleasure. And feel free to contact us, anything you may need from our side. And thank you to LIDISKI Project for giving us this great opportunity for collaboration.
All right. Thank you. Bye-bye. Bye, everyone. Yeah, bye, everyone.